Hi, I'm Kim Adams. Predictive, preventative, and curative. What do those words mean to you? Well, if you have CVD, it could mean saving your life. Right now, I have the CEO of a biosciences company called Arterez, Mike Brennan. Mike joins us now. Tell me about Arterez. What is this company?
Well, Arterez is working on both diagnostics and oral therapies. Both, we believe, will be predictive, preventive, and curative for CVD. And so it's a new paradigm.
And I should say that I didn't realize this, but 1/3 of deaths in the United States are caused by CVD.
Well, it's 1/3 of all deaths in the world. Are related to CVD.
Wow.
42% of all U.S. deaths.
So almost half of all U.S. deaths are related to or caused by CVD.
In fact, one person dies every 33 seconds as a result of CVD or a disease related to CVD.
Wow, every 33 seconds. So in the time that we're doing this interview, multiple people will have died from CVD.
That's right.
And you're developing or have developed something that may change that dramatically.
Yeah, we are in the preclinical phase of development, but we have already demonstrated that we can prevent and reverse arterial plaque in animal models and have clinically correlated biomarkers, several biomarkers that we refer to as a fingerprint for chronic disease, specifically the diseases within the CVD family.
And there's one person that is key to this research. Tell me about Dr. Tunek.
Oh, he's an amazing man. We regard him in the company as a genius. He's truly one of Michigan's best, one of the world's best and finest medical scientists. And he was born of very humble beginnings in the Philippines, in a province by the name of the Vintar province. And his parents were both school teachers, and they knew at a very early age that Joe was very bright, but also very much a teenager, and he needed guidance. So they decided that they would send him to a private school an hour away from home. By the age of 15, he knew he would become a medical scientist, and he was very hyper-focused on developing his skills as a medical scientist, even as a high school student. As a senior Ferdinand Marcos, who would later become president, named him the valedictorian of his high school, announced that he would be attending the prestigious University of Philippines, where he would get his undergraduate degree, before moving to the United States, where he began his master's work at South Dakota State University, then Penn State at the microbiology program, and then after Penn State, he moved on to the Waxman Institute, which was run by Salman Waxman, who was one of he discovered one of the early antibiotics called streptomycin. And Salman Waxman was regarded worldwide for his work that he did, the development work that he did in antibiotic research. So Joe knew that was where he needed to be, was at the Waxman Institute. As a student at the Waxman Institute, Joe would go on to develop his first drug, and he was the first student. It was unprecedented for a student to develop a drug at the Waxman Institute. And he developed a drug called hydroheptin, which is an antifungal antibiotic drug as a student. And that so impressed Merck that Merck hired Joe to be the director of research. And as director of research, Joe would go on to lead the team at Merck in the development of arvamectin, which is commonly known as HeartGuard, which is for animals, for pets.
Right.
And it became Merck's first billion-dollar drug. And then Joe would go on to develop and lead the development of other drugs across the spectrum of disease. Later, as Joe developed his career and became very well known as a medical scientist at Merck, he was then heavily recruited by Parke Davis in their later years. Parke Davis was, having some struggles, some business struggles, but they decided that they wanted to try to bring back the old glory of Parke Davis. So they hired Joe to come in and work for Parke Davis, where he would help them to develop penistatin and a number of other drugs as director of chemotherapy and antibiotic research.
Okay.
And so those drugs were commercialized to the hundreds of millions of dollars for Parke Davis.
So when he's at these big companies like Merck and Parke Davis, and he's still restricted by their own internal rules and guidelines and things like that.
Yeah, that's right.
So he wanted to break out of that.
That's right. Working for Big Pharma, it was great for Joe and his family for many, many years, but he had very different ideas, and he happened to be working alongside scientists that were developing statin drugs, and those statin drugs were very focused on cholesterol, and Joe never understood the relationship, the scientific basis for the relationship between cholesterol, or what we think of as bad cholesterol, and heart disease. He never understood that relationship, and yet these drugs that were being developed by Bart Davis, even today, are the world's leading selling statin drugs. So Joe had a very different hypothesis and theory for what the cause of chronic disease was, and he felt it was his mission to do something about that.
Okay, so he moves out on his own.
He did. He formed a company in Detroit on his own with very little money, no revenues, by the name of Firmicol. So he developed something called the Tunair Labware, which today is sold worldwide, in addition to a bioreactor, which enabled him to generate enough revenues to begin development of his first drug under Firmicol, which was called mitomycin. Once he knew that mitomycin which is an anti-cancer drug, he was able to commercialize, he then engaged people from the business community that he worked with to commercialize those drugs. And mitomycin, the first drug, mind you, that he developed as an entrepreneur, ended up exiting to a Japanese pharmaceutical for almost $1 billion.
Wow.
Those revenues then allowed Joe to continue that trend of developing more and more drugs, and to focus his energies as early as 2012 on the development of anti-embolic drugs.
Now he has this chunk of money so that he can step away and really focus on what his passion is, which is the anti-embolics. Is that correct?
That's right. He knew that there wasn't a relationship between cholesterol and heart disease, and more broadly, he was very interested in preventing and curing chronic disease. And all chronic diseases are vascular in nature. So when we talk about diabetes and arthritis and CVD, these are all interrelated diseases. And they're all complex diseases, which means they're multi-factorial in nature. Joe recognized that. So what he discovered is that at the root of all of these diseases are the damage to the endothelial glycocalyx, which is kind of the slippery lining in the artery that allows good blood flow. When that's disrupted, you're also disrupting the elements, the microorganisms that are nested within that glycocalyx, which are essential for health. Once that disruption occurs, it wreaks havoc in the vascular system, which then leads to multiple chronic diseases.
So it's like upsetting your internal ecosystem, really.
That's exactly what it is. It's very well said. And so what Joe recognized or identified as the root causes, the multifactorial root causes of heart disease, were the disruption to the glycocalyx, inflammation, and oxidation. So what he did is he began to look at the biomarkers, the associated biomarkers that are shed from the glycocalyx, to see if he could clinically correlate those biomarkers with the shedding of the glycocalyx as it relates to various CVD diagnosis. And in fact, he was able to do that. Now, this was in 2011 and 2012, before anybody was talking about the glycocalyx.
Okay.
And so that's when it all started. And yeah, you referenced the anti-embolic drugs. This is a very similar to antibiotics. Antibiotics was the discovery of microorganisms at the root or at the cause of infectious disease. We believe that the glycocalyx is at the root of chronic disease.
Okay.
And that's where this all began.
So he had to really turn what we had known upside down, which is that cholesterol, we would think, and we've been told, is the cause. And so we have all these medications that we're using to treat high cholesterol and changing our diets. And that may not be, according to Dr. Tunick, that's not the cause of it.
Well, I mean, having a good diet is always a good idea. A healthy blood flow is, of course, essential to good health.
Okay.
And so there's certainly Dr. Tunick wouldn't argue that it's okay to have a bad diet or run out and eat McDonald's every day.
Right.
But what he would argue is that reducing cholesterol is pointless. It's not a good idea to do that at all. In fact, cholesterol is essential for good health and well-being. It's a healing agent in the body. The very first cholesterol drug that was created for a disease called familial hypercholesterolemia, which is the excess creation of cholesterol in the body. It's when the liver just can't stop creating cholesterol.
Okay.
And that can be very dangerous. It's very bad for your health. And so a drug called lovastatin was developed to address familial hypercholesterolemia and proved that it could in fact reduce cholesterol for those patients impacted with this hyper condition.
So it was created for a very specific group of people
That's
with
right.
a very specific disease, but then somebody went, oh, so it reduces cholesterol.
I wish I could argue that that's not what happened, but that is what happened. That's right. That's exactly what happened. Not only that,
really.
then from there, the lipid panel was created. And the lipid panel, which today doctors measure you by your cholesterol level, known as the lipid panel, right? Well, that's what a lipid panel is. It's measuring your cholesterol level. That was measured, that was created for lovastatin. But now the lipid panel is used by cardiologists widely to measure your cholesterol. And every year, they reduce the recommended cholesterol level. In the 1960s and 70s, a cholesterol of 400 or 450 was normal. When you cut off the pathway or the cell's ability to biosynthesize cholesterol, you also cut off the body's ability to biosynthesize CoQ10, which is essential for heart muscle.
Okay.
And your heart muscle works 24 hours a day.
Right.
It requires CoQ10. So it's a really bad idea.
But so what's the solution then? Because people are doing just that. They're taking medications to lower their cholesterol.
That's right. Well, we believe the solution is a new paradigm.
Okay.
Certainly a healthy diet, exercise, a healthy lifestyle is a good idea. We know that smoking and any foreign particulate that enters the bloodstream will damage the glycocalyx, will do damage to the heart.
Right.
So none of the kind of fundamental lifestyle concerns that we have today, we're not suggesting that any of that should change or that any of that is wrong.
Right.
What we are suggesting is that the body creates the cholesterol that it needs to use. In fact, the cholesterol you ingest only has a minor impact on the cholesterol levels in your body.
Right, which is why people who may have a very clean diet still genetically have high cholesterol.
That's right. And when we look at the data related to statin drugs and the reduction in death rates on an annual basis, what Dr. Joe theorizes is there's a lot of confusion about that. Each statin has other ingredients, such as anti-inflammatories, that may in fact be providing the ancillary benefit to the patient, whereas the statin itself is not providing that benefit.
Okay.
That's right. So you asked the question, what is the solution? Well, the solution, we believe, is to focus on the root causes. What are the multifactorial root causes? As I mentioned, the repair to the endothelial glycocalyx, inflammation, reducing inflammation, and oxidation. Those are the three things that we're focused on. So what Joe did was he developed 8 compounds and tested each of those compounds in various combinations until he saw positives across the board relative to preventative and curative.
Okay.
He then also created an animal model. And this animal model, the animal model that's used today for atherosclerosis, for the study of heart disease, is known as the ApoE mouse or knockout mice.
Okay.
And the reason that they call it the knockout mice is because what they did is they limited the mouse's ability to create a biosynthesize a protein that allows for the natural movement of cholesterol into the bile.
Okay.
So that you prevent the mouse from being able to biosynthesize cholesterol, to move cholesterol. So instead you get the accumulation of cholesterol in the mouse artery, which is false. It's not the way plaque is created in a human being. So we're using an ApoE model with a buildup of cholesterol, developing drugs to reduce that cholesterol, testing it on animals, and showing that we can reduce cholesterol in animals, which does result in reducing cholesterol in humans, but has nothing to do with heart disease.
With the heart disease, right?
Yeah.
Okay. So where is Dr. Tunick in his research now? What is going on now?
Well, we're further developing what we refer to as the Tunick arterial plaque natural mouse.
Okay.
And what this means is we're now using MRI and other translational technologies so that we can move from animals into human beings.
Right.
So our focus, our objective as a company is to get into human trials against hypertension, heart failure, and CHD, against plaque. So we can look at and identify plaque reduction in human beings, the elimination or reduction of hypertension, and improve blood flow dynamics in heart failure patients, which could very well extend life.
So what's it going to take to move? So the mouse has not been developed yet completely, correct?
No, the mouse has developed and has been proven using a technique called histopathology, which is the sectioning of mice.
Okay.
What we want to do, which is groundbreaking, is to use MRI technologies, very high resolution, to look at the coronary and various parts of the mouse heart where we're naturally creating plaque.
Okay.
And then seeing how our drug prevents and reverses that plaque from occurring.
And then you can move into human trials from there.
And then move into human trials, right. So
is there a time frame for this that you're thinking?
Yeah, we think we'll be in human trials by 2021 at this point. Our 8 compounds have been fully synthesized and developed.
Okay.
We're now producing those to just below GMP standard, which is the standard for human beings would be GMP. We'll then develop to GMP standard, the drugs to GMP standard. And we're also continuing the development of our diagnostics with human beings, where we've already demonstrated that we can discern the difference between various diseases, which again is groundbreaking on the diagnostic side. So effectively, we believe we'll be able to diagnose or predict what kind of disease you have, what kind of chronic disease you have, and within the CVD family, what type of CVD disease you're likely to have.
Well, that's the part that fascinates me. So I understand the curative aspect of it, and even somewhat the predictive, but it's the preventative, that if you're developing something that could actually prevent me from getting it in the first place,
right,
that to me is incredible.
It is, and we're really excited about developing the diagnostic, but as we often say, what's the point of being able to diagnose something if you can't do anything
about it? Right.
And so that's why we're focused on both the diagnostics and the drugs. And it's very important that we have this triple compound oral therapy, which we believe will be water soluble, non-toxic to the liver, which will allow the prevention and reversal of arterial plaque, thus anti-embolic mechanism.
So when you say predictive, how do you envision that happening? Am I going in for a blood test?
Yeah, that's a great question.
How do you
It would in fact be a blood test.
Okay.
Now when the doctor sends out that blood test, that blood will be put through one of our labs through a proprietary algorithm, an Arterez proprietary algorithm that will help the physician to discern which disease they're looking at by our fingerprint technology.
And would it be your, would you be able to tell me that I am going to get it or that I could get it?
Yeah, that you would be at risk for it.
I would be at risk.
But it depends. It depends on where your markers are and how far this technology advances. We may in fact be able to diagnose these diseases and various stages of the disease. So for instance, we think we may be able to discern the difference between what's known as ischemic and non-ischemic heart failure, that is with plaque and without plaque, which would be a wonderful find for emergency room physicians. When they have somebody come to the emergency room, they're unable to discern that. Right now, if you have a heart attack, you have to have a heart attack first before they look for the troponin level. Well, troponin is just one marker. Imagine a world where we could use many markers as a means to determine what you're at risk for and what you have. And if you have it, hopefully you've been diagnosed at a very early stage.
That's what I was going to say. So it's
that
like
you can
early
do something
detection,
about
right?
it. That's right.
So, and it would be up to the patient, you know, they would know how many markers they have. So I may likely get it, so you would put me on medication?
Well, we wouldn't, but what the doctor may make some recommendations. Now, those recommendations could be as simple as lifestyle changes, or as we further develop our anti-embolic drugs for treatment, then those may be recommended. And keep in mind that our predictive diagnostics could be used for our anti-embolic drugs or any drug that's developed in the world that addresses the endothelial glycocalyx at the root of chronic disease.
Moving forward, where do you see the overall value of the company? I mean, is this the next antibiotic?
You know, we really think it could be. We really think so. We see this as the next paradigm, or as a paradigm shift, and this new platform of predictive, preventative, and curative really being where the industry is going to go. You know, medical science moves very quickly. And if you were to ask a cardiologist today if he would use the same treatments that he used 5 years ago in a patient, they would tell you absolutely not. So every 5 years, the industry transforms itself. And so we think this is the next iteration, and that will play a major role.
And you think within that time frame of 5 years, or what is your company's time frame?
Well, we're looking to license and or work with larger biopharmaceuticals in the furtherance of this development and commercialization of our diagnostic and oral therapies right out of human trials. That's our goal. So by 2021, or certainly early 2022, we think we'll be exiting or working cooperatively in a co-development relationship with one or more of the majors in development of our technologies.
And so for someone that wants more, we've covered a lot, obviously, and you know, to get really into detail, you do have an amazing website that people can go to and really dive a little deeper into what we've already talked about.
Yeah, you know, because of the complexity of our sciences, what we took the time to do was to develop a 90-minute video. It's chaptered, of course, so that an investor or viewer interested in these topics could look at, you know, watch various chapters, whether it's the history of cholesterol and statin drugs, or the multi-factorial root causes of heart disease, or what we're doing about them with our diagnostics and oral therapies. And that video is available on our website, arterez.com.
So it's Arterez, A-R-T-E-R-E-Z. Arterez?
A-R-T-E-R-E-Z.com.
Perfect.
That's right.
Great. Great name.
All right, thank you.
All right, thank you so much.
Thank you.